honorable and respectable teachers, honorable respectable directors from different institutes, respected physicians, doctors, postgraduate students, as well as the gentlemen, ladies, also the delegates from the home and abroad. First of all, thank you very much, the Eskayef, for bringing me on this historic day of Bangladesh as well. I'm really delighted to have this opportunity created by the Eskayef Pharmaceuticals to give a delivery on the part of Monuvir, which is one of the important historic revolution, I would say, that in the last 2 years. Gentlemen, ladies, we know the COVID-19 situation, the pandemic has been well controlled and stable in our country. But remember, the game is not over. If you follow the pandemic situations around the globe, surrounding the Bangladesh situation, even in the Southeast Asia region, as well as Europe and Western area, the situation is not very much stable. There is every chance that clusters are ever there, and there is still chances of developing the new variants, and which can create a problem in different countries as well. That includes the Bangladesh. So even though we are in a stable condition, the situations that can create a problem in the future means that scientific evolution and revolution must be carried on. And through this endeavor, I would be saying that it's a great opportunity for us to have our first oral anti-COVID drug in our hand. And so much so quickly that, again, I'd like to give thank you, Eskayef, for giving this wonderful drug to our people of Republic of Bangladesh. So I will try to give something highlights on the part of Molnupiravir, which is said to be the game changer in COVID-19. As I go through many of the articles, I would like to say that we'll be giving this background information of the Molnupiravir, how it develops. I will try to give some of the picture, how the mechanisms of the drug action happens in our body, which will be a little bit of tough. Thirdly, I will try to give how this trials has been going on in the animal studies, also the human studies in the phase 1, phase 2, and phase 3. Few of the interim analysis I'd like to share with you, and later on, the recommendation that has been given, even though there is no still authorization, just an emergency use of authorization has been provided. So, gentlemen, ladies, with no more waiting, let's start. So, Molnupiravir, the game changer in COVID-19. You see, this is the common scenario that has been situation in all around the globe. On the right side, it may be in Southeast Asia regional, low molecular income country hospital, or maybe on the left side, you can see it may be a very advanced case of hospital in the United States of America. Wherever it is, the patients are worst, sometimes severe to critical, having a COVID-19, and some of them has a poor outcome in the form of the death. The issue is that science never rests when such situation happens. So you can see the search where the scientist is going on, and right now the concept is hit early, hit hard. That's the conception, because we did not find a magic things which can prevent the severity. Still, after these 2 years of huge amount of searching is going on, we're still not sure which of the patient is going to be in severe or critical, which of the patient can have spontaneous recovery. Although there has been some of these risk factors like the older age, maybe diabetes mellitus, hypertension, and even though there may be so much of beauty that risk factor can be biased by the physicians that have said it. But this drug, which has given some initial hope in the name of the remdesivir, and then came the big revolution in the name of the dexamethasone when it comes up as an immunosuppressive agent, because it has been found that this virus has an interim inflammation, and this needs to be stopped at some of the stage when the severity pulls back. And when this dexamethasone came up in the form of either tablet or injection, it revolutionized the treatment because it reduced the mortality, obvious in COVID-19. But since then, what happened? Let's see. So the current approval treatment for the COVID-19 as an antiviral issue was not very much huge. You can see it was started from the remdesivir, which was injectable and IV infusion. And problem is that it was not consistently, and there was a variable responses. Some of the response was very good, especially if you follow this active trial in the United States of America. But uniformly, there was no constant presentations of the efficacy of this remdesivir all through the patients of the COVID-19. And that is the reason why WHO could not recommend it as a drug for choice. Then we have a tocilizumab. Initially, there was so much controversial issues about Actemra, but it was later proved that it severe and critical illness, especially when the progressive hypoxia is present, the tocilizumab reduced the mortality. And last of all, this is the polygeneron. We all know that casirivimab and imdevimab. This combination gives a little bit of hope because this is the agent which has been proven that it may reduce the severity of illness if we can provide it early. Big problem is that all of the agent that has been described is injectable form. It needs supervisions, you need to give to the hospital settings, and it needs expertise to give these drugs for any kind of amount. So this is a big problem because the patient who remains in the milder illness or the moderate type of illness can remain in the home, and they will not go to the hospital settings even if the pandemic is going on. So how to resolve this? And the results came out in the form of this oral drug of Molnupiravir. You can see the Medicine and Healthcare Products Regulatory Agency of the United Kingdom authorized this one, Molnupiravir, for the treatment of mild to moderate COVID-19. Again, I repeat, this is the mild to moderate COVID-19. So this is the highlighting news. And also we find that it says that the Dr. Leon Retain is the MHRA chief executive. But the Molnupiravir is the world's first approved oral antiviral for COVID-19 and can be administered outside of the hospital setting, which is very important. It's an oral drug, we can use it even in the home. And then the chair of the Commission of the Human Medicine, Professor Sir Munir, you can see on the right side in his faces, with a big smile, said that he was found to be effective in reducing the risk of hospitalization or death in non-hospitalized adults with mild to moderate COVID-19 by 50%. So that was the issue why it becomes a big magic, as a name as the magic bullet. Well, Sajid Javid, it has been shown by you as well. The UHA authorization for this antiviral is an excellent addition to our memory against the COVID-19 and will be also a game changer for the most vulnerable and immunosuppressed who will now receive a groundbreaking treatment. So that was the statement Sajid Javid given when the launching of this program happens as an emergency use authorization. All are good news, but let's start about the Molnupiravir. So it looks like this. You can easily see this look like this structure. And it's orally available, but it's not a drug, it's a prodrug. And it has a nucleoside analog and hydroxycytidine. This is the main agents which is active within the body. The interesting point is that it exists in the two tautomeric forms, in the form of cytosine uracil nucleotide, and that exerts actually the antiviral effect. Now, the big issue is that when we are considering whether ribonucleic acid and other things, you know that there are 4 bases: adenine, cytosine, guanine, uracil. So I will be giving some of the picture how this drug really acts. Maybe it will be difficult to understand by all, but let me give a simple try. So these are the indications that actually provided. Right now we are giving it for the COVID-19 in the situations. But this is not the drug that has been given in the last 1 year, 2 months issue. It has been there in the pipeline since 2013. It was tried in the influenza, many other coronaviruses, not only the COVID-19 or SARS-CoV-2, but other seasonal coronavirus also has been tried by this Molnupiravir. Encephalitis alphavirus in the form of Eastern, Western, and Venezuelan equid virus, and also the hepatitis C. So there are many attempts of this virus, antiviral drug, many of the viral agents. This has been initially used for the influenza. Later on, the repurposing happened for the other viruses as well. So let's have some discovery fact. You can see the identification has been done in the form of the EIDD. EIDD means the Emory University, which initially started about the Institute of Drug Development 1931 as a replication. It was in 2013. In 2018, the Molnupiravir actually overcome the bioavailability issue. In the December of the 2019, there was an emergence of the COVID-19, as you all know that in the China. In 2020 January, the Emory University, which is the Institute of Drug Development, planned the submission of the investigational new drug, Molnupiravir, to treat the influenza. I told you this was started for the influenza. And even in the 2016-17, when the Ebola gives a crisis around the globe, the Molnupiravir was a trying effect. In the February, US FDA actually asked the Emory University to furnish all information about the Molnupiravir against the pathogenic coronaviruses. In the March, the filing of the influenza investigational drug and licensing deal with the Ridgeback Therapeutics. This is the one small biotech company in the United States who initially came up with this one. In the April, the Molnupiravir was licensed to the Ridgeback Therapeutics. In the May, Merck actually came up later on, acquired the exclusive worldwide right because they're a big company. And if you want to make drugs available all through the globe, we need to have a large company to come out. The Merck came up. In the August, you can see how quickly things are going on for the phase 1 clinical trial completed within the human. In the 2021, February, phase 2 clinical trial was completed. In the August, the phase 3 clinical trial started. And in this interim period of time, we already have few of the results of the phase 3 clinical trial. So you can see the approval came up at the November 2021. So within a very short period of time, the development was very crucial because we are very need to have a magic bullets. We do not have any magic bullets since this year's November came up. Before saying something about how Molnupiravir works, I'd like to give a highlighting point to you, how the virus actually causes replication in our body. We all know a little bit of it, but let's have some recap. We all know something about the spike protein, which actually combined with the ACE2 receptor. But let me tell you something, when they cannot combine, evacuation happens within the cell membrane. And after that, a trafficking happens with the incorporation of the cell surface of the virus. This surface is come from the cell surface of our body. After that, you can see there's a membrane fusion happens. Why this needed? This is needed because we want to release the virus particle to the cytoplasm. If we do not have the coating viruses, we will not able to release it to our cytoplasm. So you can see this is the genome of these SARS-CoV-2 viruses. But the big issue is that it needs to be replicated if you want to have a lot of viruses in our body and cell. So what will happen? It happens that the initial translation of this genomic virus happen and it creates lots of polyprotein. Look at this polyprotein, 1A and 1AB. So why these are needed? This is needed because we want to break this protein by a cleavage to have a large number of the non-structural protein. So you can see a lot of colors are there with the non-structural protein. And this actually is very crucial as because these are named as the RNA-dependent RNA polymerase. Or we can also say it's a replicase because we want to replicate the virus. We want a replicase enzyme. This is the enzyme. So you can see on the template of this virus genome, these non-structural protein, look at this one, 8, 10, 13, 14, there are 16 non-structural protein. And with this template, it actually causes the transcription to develop many of the important genes that is needed for the virus. So you can easily see virus is using our own machinery to develop its own viral particle. And this subgenomic known as the nucleocapsid, look at this one, spike, membrane, envelope. All the genomic tooling has been done through the process where the non-structural protein is very crucial. And after that, what happened that the translation happened in our cell's endoplasmic reticulum. Virions, then formation happened, combination, and then virion get a release after taking this cell membrane surface and go on. From one, you can have developed hundreds, thousands, millions, trillions, and so the devastation goes on and on. So this is the basic mechanism how the things happens. And I will give you a non-structural protein, huge number. I told you there are 16 non-structural proteins out there. These are very crucial part when we are doing the replication of the virus in our body. Now, when the replication happens in any viral genome, there's every chance that there will be mutation happens. And if the mutation happens, it has an intrinsic capacity to correct it. This is called the proofreading. And that is the reason why I told you the proofreading. So the proofreading causes the corrections of the genomic virus so that it can replicate of its own. Now let's come to the part of how Molnupiravir works. We told you Molnupiravir works on the part of RNA-dependent RNA polymerase. But so also by means of remdesivir, so also the favipiravir. So what's the difference then? So this is the difference that I'd like to tell you. Now when Molnupiravir actually acts as a prodrug, it combines in our body and leading to the N-hydroxycytidine. This then combined through a kinase known as the NHC triphosphate or Molnupiravir triphosphate. What I like to mention you that when Molnupiravir triphosphate happens, it in the part of the genome just incorporate the M instead of the A. So, you know the basic tool of the nucleotide is the A, G, T, and C. This is the nucleoside that is needed for developing the genome. All they have to do is just replace it through the M, T, P. So you can easily see that cytosine will be converted to uracil, and some of the adenine will be converted to G. So when this thing happens, the mutation will try to correct it by proofreading. But this is going to be happen again in the next cycle, again in the next cycle, and again and again. So you can easily see there are so many errors that happen in the virus genome. That virus could not correct it, cannot proofread this thing. They become hustle and bustle, and so that at every cycle generation, mutagenesis increases and increases, and leading to the serious amount of catastrophe. So that is the reason why they call it error catastrophe. So what happened that through the mutagenesis two-step process, this molnupiravir not only causes the problem of the development of replication, but also inhibits the viral capability to correct the proofreading. That leads to the serious amount of virus, and virus dies. This is how the mechanisms of unity for the molnupiravir. But what remdesivir has been doing is that they could not make so many errors. All they have to do is to make the chain termination early. So it cannot causes the problem of the mutation and causing the virus to hustle puzzle, just slow the rate of the chain termination. So that's the basic difference between the remdesivir and the molnupiravir. So you can see this two-step mutagenesis that I've been telling you. So in the first step, the enzyme synthesis negative stem actually is become problematic. And the second step, again the subgenomic RNA is synthesized by M-containing RNA, which is causing more and more mutagenesis responsible. So this is how it works. And this is the evidences of the papers that has been, I've told you about this molecular cell paper where the mechanisms, differences between the molnupiravir, the remdesivir, and as well as favipiravir is there. Although favipiravir has the capacity to go for some of the mutation and chain termination, the big problem with the favipiravir is that it has a very poor pharmacokinetic property. There are so many problems in the heart as well as the liver and kidneys. Remdesivir has also its limitation on the part of pharmacokinetics, as we all know. We cannot provide it to the part of the renal problems because it causes serious AKI sometimes. The big issue is that molnupiravir has a wonderful pharmacokinetic property. There are less amount of side effects that I will show you in subsequent slides. So this is the paper that actually shows how the mechanism differs. This is another picture that I have taken up to show you wonderfully how it beautifully, this surprising G and M actually gets involved and replaces the normal cytidine of this virus messenger RNA. So you can see whenever there is incorporation M happens, it causes mutagenesis. On the right side, you can see the mutagenesis, which leads to huge amount of error, which the virus cannot correct. So let's come to the trials. I mean, when it is given to the animal, they choose one of the animal known as ferret. Now, when the ferret animal is chosen, because it can cause a mild to moderate illness, not to severity. It does not cause the lung involvement, because we want to choose those person who is having a mild to moderate illness, not the severe and critical. The problem with the mice and mouse is that whenever somebody given the virus inoculation, it quickly can transform into lung. So that's not our subject of interest. That is the reason why the ferret was chosen. And you can see these, this is the control group, and this is the group where the drug has been chosen to be given. Initially, through their nose, huge amount of virus loading has been done, and thereafter this drug is given as an oral pellet to check whether this drug can prevent the severity. And secondly, they want to also show whether the other contact animals get the infection or not. And thirdly, they want to see whether the drug is safe or not. So these are the things that has been shown in the printing. Look at this one. When the drug is provided, within 12 hours, the virus load reduces tremendously. It just came from the 10 to the power 6 to the 10 to the power 1 within the 12 to 24 hours. So that was the surprising thing, that the compound concentration happened. But look at these concentrations in the control group. It remains over there, while the baseline has been touched within the 24-hour time. And it's been saying that almost all the virus RNA particles has been cleared up within 24 hours duration of time after giving these oral drugs. As because when the oral drug is given, the absorption was pretty quick, the pharmacokinetic property is very nice, the concentration jump up very quickly, and also the mechanism of action. So you can easily see this is the drug that is given this small dosage. This is another dosage, the red one, and they also see the responses. And you can see the response over here. You see the control has the 10 to the power 4 virus particle remains over there, but look at this one. When the drug is given, it just goes to the baseline. So that was the reason why the animal study was so much successful to be seen. And when the adversity has been shown, there was a very less adversity been found in this ferret. Not all of the ferrets actually dies. Even the control group, there was a severity, but there was no death. So let's get into the human trial, and it started with the phase 1. As you can see, to check whether the patient is safe or not, because phase 1 trial always believes safety first. And the side effect profile was, most of them have a mild grade 1 reaction. And the percentage of the reactions to the mild, moderate, severe was same in the molnupiravir group as well as the placebo group. Even 1600 milligram was tried with a single bolus dose to check whether it is consistent with the phase 1 study of the human or not. And it came up good. So emergence adverse event was also check 1, and look at this number, 1, 1, and 2, and 3. Even the 300 milligram to 800 milligram was tried. And interestingly, severe adverse reaction, serious adverse reaction was found in 4 patients. Among them, one of the patients dies who was receiving the placebo. There was no death happens when the patient receiving the molnupiravir, even though patient have severe illness. This is another study of the phase 1 to see the safety of molnupiravir in patients with early SARS-CoV-2. Phase 1 open level dose escalating. This study was based on whether how much doses we should be given, 200, 400, 800. And it came up that there was a tremendous benefit happens when the 800 milligram was chosen. And look at this one, side effect, cardiac, gastrointestinal, general, infectious, investigation, musculoskeletal, nervous system, remains 0, 1, 0, 0, 0, 0, everything. So you can see the organ-based serious adverse reaction was basically absent. An interesting point is that when 800 milligram was chosen, it was found to have good profiles on the side effect rather than 400 milligram, 300 milligram. And that is the reason why the dosage has been selected as 800 milligram. So this is the phase 2 study. And this has been published recently in the Matrix, which I have taken. 240 has the availability, and it was divided 200 milligram molnupiravir in 23 patients, 62 for 400, and 55 with 800, and placebo 62. So it's very stringent enrollment, allocation, follow-up, and analysis was done in this randomized control trial. What about the results? You can easily see visit was given in the day 1, day 3, and day 5 because the 5 days duration of treatment was provided. And when we're choosing about the placebo, the red number indicate the positivity of the viral in the day 5, day 3, and day 1. You see 200 milligram, 400 milligram, 800 milligram. Look at this 800 milligram. You can see there's a complete negativity in the day 5. Even in the day 3, it was only 1.9%. Look at the 400 milligrams virus particle who are also there. And look at this one. When we are considering about the dosage, drogas, and cumulative percentage of the responses, that was tremendously excellent when the 800 milligram molnupiravir was given. So the phase 2 trial actually shows not the safety issue, but the efficacy of a drug of the molnupiravir in the human trial. About the adverse event, you can easily see the percentage positivity about the control and the different groups of the drug was remains same almost, 29% to 35%. It remains same, and there was a very less amount of adverse event. In the name of the dizziness, insomnia, some of the patients developed the diarrhea, and few of them developed the skin rashes. One of the patients has developed hives. That has been, I'm telling you. Let's come to the phase 3 trial. Although there has not been any published article over there, but the interim data as well as other things has been came up, which actually shows that against the placebo, day 3 and day 5, the molnupiravir action, you can easily see that day 5, 0% COVID was positive at 200, 400, and 800 milligram. But it remains 224% positivity within this part of placebo. So what was the result actually? The result came up that 7.3% versus 14.1%. there was no death in the Molnupiravir group, but in the placebo, we found that there was 8 deaths happened. So at day 5, when the RT-PCR test was done, it was almost 0 when the Molnupiravir was chosen, but it was around 24% when the COVID positive was there. But remember one important point that in this study, what is the remarkable thing is that when the RT-PCR was done, they have chosen also to see the genomic sequencing, because we need to sure, is it the replicating virus still there or just an RT-PCR positive? Because we all know that RT-PCR does not necessarily means the infectivity. There may be viable virus and there may be non-viable virus. The good news is that this study actually shows whether the virus replicating virus or not. And it becomes 0. So that's the best part of this paper. So what was the extraordinary thing that has been shown? It's shown that it has 50% reduction of the hospitalization or death. So that's the magic that we are looking for. Is there any drug, if we give it in the mild case, can reduce the hospitalization? Is there any drug, if we use in the mild to moderate case, can reduce the severity or the critical illness? It causes 50% reduction. That's wonderful. And there was no death in the study. At the same time, there was 8 deaths in the placebo control. We told you. Now, what I'm going to show you that has not been published, not been mentioned, few of the study done in India. And in that Indian study, also the clinical parameters shown to be seen. That's because we are all clinicians over here, most of them. We want to check whether the Molnupiravir has any clinical improvement or not against the standard of care. It was 63.4% versus 22.3 on day 5. On day 79 versus 49. As the days goes on the second week, we did not get too much of the variations between the Molnupiravir. So it has to be given early, because the tremendous benefit was found in the day 5, you can see. And not only for that, the faster time for the clinical improvement, Molnupiravir versus standard of care shown that 8 days versus 12 days. Look at the p-value. Not only that, whether it can cause us more RT-PCR negativity, yes, it is 77.4% on the day 5 versus 27.1%. And also the 94 versus 57 for day 10. So if you can provide it within the 5 days duration of time, there's every chance that you can reduce the RT-PCR negativity. It can improve the clinical improvement and also the faster time for the clinical improvement. Another study that has been in the phase 3 trial shows that the Molnupiravir versus standard of care, 78.3% versus 48.4. On the day 5, it becomes a big difference. But when it becomes day 14, look at this one, 99 versus 98. So you are not able to give this drug when the patient is severe. You will not able to give this drug when the patient is taking a lot of oxygen. You cannot. Because there's no difference has been found between the placebo. So the selection of the patient is so much important. Otherwise, we will be in deep trouble. Faster clinical time also seen the day 5, wonderful. It was around 80% versus 50%. Now, futility. What I'd like to mention that there was a few studies done in the Indians in the phase 3 trial has the futility. Why? Because the patient was given a severe and hospitalized. There was a poor selection of the moderate to severe group. And those patients, when Molnupiravir was given, have a more death. So that is the reason why you are going to be deteriorated patient if you select to give it in the severe to critical patient. Futility studies are there. And also there are so many ongoing studies. There are 5 of them, 10 of them. So this is going on. There's no interim results we have been found about this one, but the assessment is still going on. Hopefully we will get it in due time. So 4 facts about the Molnupiravir is that there was a 0 death in the treatment group when the phase 3 is concerned. It has to be given within the 5 days of symptom. 10% patient treatment and placebo group reporting in the side effect only. 1% treatment and 3% the placebo group discontinued the drug because of the side effect, and the number needed to treatment is 15. Means that you need to treat 15 patients to avoid 1 hospitalization. You need to give 15 patient treatment by this mild to moderate case to reduce 1 death in the Molnupiravir. Does that exist all of the variants? It does. You can see the gamma, delta, mu viral variants in clinical trials conducted globally more than 170 clinical trials, and it has been found to be effective. So how to take it? I think we have been discussing it. The 800 milligram is the ideal dose, so it has to be taken 200 milligram tablets, 4 plus 0 plus 4. Not only the mark, we also have Monuvir in the name of Eskayef. Thank you very much again bringing this wonderful drug. Side effects, as I told you, sometimes you can have a dizziness, you can see, and sometimes people may have hype, but fortunately there was no fatality related to the side effects. Most of them are milder. Pregnancy, I'm afraid we are not going to give it any drug to this pregnancy because there was no study. Can we give it to the children or below 18? I'm afraid not, because we don't have any study. All the study that has been provided in the phase 1, 2, and 3 trial is given more than 80 years of age. So unfortunately, we are not doing. About the future plan, now the Mark has actually planned to give 10 million courses of treatment at the end of 2021, with more doses expected in 2022. We are expecting it. And the US has already agreed to deal for 1.7 million courses. So these are the huge number of crews that you can see in these online versions, those who are listening also seeing. And all of them are very much enthusiastic about the effects of the Molnupiravir that has been shown in the different trials. It's a game changer. Does we have the magic bullets in hand? Can we use it? Can we use it prophylactically? So many questions are actually coming up hugely to this interim results of clinical items. Now, what I'm going to show you that has not been published is a meta-analysis that has been done on the Molnupiravir. Even though we do not have a larger, huge number of trials, the amount of study that has been provided gives a meta-analysis. And you can see the Fisher, Hetero, Optimus, and Ku and Mark, all of them are randomized control trial. And about this one, we found that about thousands of the patients, enrollment and the control was 995. The 54% shows their clinical improvement. And among the all studies we consider, there's a 54% improvement was seen. All the lines were on the left side means that Molnupiravir does have favors rather than the favor in the control. What about the meta-analysis of the improvement on the part of hospitalization? You can see there's a 57% improvement. About the study of Fisher and Hetero and all studies. So there's a serious amount of improvement of hospitalization prevention. What about the improvement in the part of early treatment? You can see the 89% improvement has been observed in the favors of Molnupiravir. So if you can provide the treatment within the 5 days of symptom, remember, within the 5 days of symptom, then you can have an 89% improvement in this meta-analysis. So you can see the number of trials and Fishers and the improvement lists. I'm not going to exhaust you anymore. And it shows that green lines are actually more and more in the left side. And this is the baseline of the forest plot. So that gives it tremendously favors in benefit in the form of Molnupiravir. So now what happened, Peter Horby, who is a professor of the University of Oxford, saying, he said that proportional reduction of the risk of hospitalization death is impressive. But it is important to remember that the absolute risk were 14% reduced to 7%. So quite a lot of people need to be treated to prevent one hospitalization or death. This means the drug needs to be very safe and affordable. The safety that also looks good. So again, this is encouraging. A virologist from the Vanderbilt University, the Nashville, Tennessee, we are very happy that one of the renowned molecular biologists from Vanderbilt University, Dr. Tanim Amin, is also with us. And he is also saying that this virologist said that I was pretty blown out by this one. Not in this year, he said it in the 2015. The ability of the virus have a so much unique that it can capable to say it's just blown away by any other drugs. And lastly, William Schaffner, he's the one who is the key person for the Moderna vaccine, you all know. He said if the government controls the distribution, then it may be more restricted, like the polygeneral and monoclonal antibody. But if left to the physician only, then it may be more widely, even in the beginning, it can be used. So monoclonal antibody are given via infusion or subcutaneous injection and require professionals actually to administer it. That's why oral Molnupiravir has the potential to be a game changer. So the key message, gentlemen, ladies, number needed to treatment for Molnupiravir is 15. So the drug needs to be very safe because we need to give 15 patient treatment to get one benefit. Molnupiravir has to be used early, less than 5 days of symptom onset. Again, I'm saying, do not use it in asymptomatic patient. Before someone is deemed ill, enough when it don't work. Thirdly, the older age with at least one risk factor with mild to moderate illness at home is the ideal candidate to receive the Molnupiravir. Next, lack of any study that is evaluating the role of Molnupiravir in the breakthrough infection following vaccination, because it is going on all through the world. We need to have further study to establish whether it interferes with available vaccines or not. Cost effectiveness and accessibility for all is a major issue that need to be dealt with, and the effect of Molnupiravir with other therapy for COVID-19 needs to also to be examined. So that's all. Any questions?